So 1 millimeter to the right, there are his brothers of the same colony. 1 millimeter to the left, there are his brothers from a different colony. How does he recognize? How does he know that these guys are from the same colony and those guys are from a different colony? So we know some of the mechanisms for protein recognition and these sort of things, but the more important question is why? Why do they need to recognize? If we have two colonies from the exact same strain, why would they not just merge to one big colony? Why remain separate? So we have the why and we have the how. And these are really big questions, and they have a lot of application for agriculture, for medicine, for antibiotics, for all of these things. And a lot of people are researching on how bacteria recognize self and non-self.
So, what's the possible case for us to study this case?
What is the what?
If you want to study this case, start to study this case.
Oh, well, first of all, you would make experiments like which bacteria recognize each other. Like if we have one colony and second colony, and they have, like you see, they have a boundary between them. So how different they are genetically. So you would sequence the whole genome of this bacterium and the whole genome of this bacterium, and you will see how many differences they have. And you can extract proteins. So we know that there are many proteins that are active in recognition of other things in the environment and fighting other things in the environment. So you can start to look at which proteins do they have. Like we have in our immune system, we have proteins which are, their job is to recognize cells that are from us and cells that are from other people. So we can look at the cellular level. Are there things on the cellular level, like in our blood or in our system, that make them recognizable for each specific colony? And so we look in the cellular level, in the microscopic level, in genetic level, in protein level, and we can study the surface of the cells to see which do they have something like antigens, like we have, that make them recognizable. We can create differences in colonies. So we can take the same colony and we can take one cell from this colony and make a tiny mutation that it's really 99.9999999% the same, but there's one mutation. And we can see, do they still recognize each other? And then we make two mutations. See, do they still recognize each other? And then we can see what is the limit of how much can I change the genome and still remain myself, and how much can I change it and then suddenly I'm someone else. And we can do that with different genes, so we know which genes are responsible for this recognition. So what I've said now is maybe 2 or 3 PhDs.
Yeah.
Oh, wow!
This is from a lady who's working in her project. Here.
Yes.
It's for the anti-fungi capacity of actinomyces.
Right.
Yeah, and then she showed that this is here. There's not much of the composition.
So this is the fungi.
Yeah, this is the
And
the central.
Yeah,
This is the actinomyces.
yeah,
Actinomyces
and then yeah,
to see.
and this is
This is the
fungi.
fungi.
Yeah, but with here it's like they have the clear border.
Yeah.
Here it's not very clear.
Nice. And this is streptomyces.
No,
No.
actinomyces.
Yeah.
Streptomyces.
Yeah,
Streptomyces.
she's here.
Streptomyces.
Yes.
Yes. This is usually streptomyces is the hero of the antibiotics game. Perfect.
But in this
So-
case,
What?
he's a hero.
Yes. Yes. So now you can take this bacterium and start applying it for antifungal materials. Look, what does it do?
But in this case, can we connect this anti-resin?
Yes. I mean, if I don't know if they are not touching each other. Yeah, it's probably. You can see here.
Yeah. And this.
You can see here there is a lot of growth,
Yeah.
and here also, but here and here
Yeah.
there's not so much growth. So
Maybe.
yeah, or maybe the fungus. The fungus has anti-streptomyces, antimicrobial, antibacterial.
Oh,
Because also fungi have a lot
so
of.
we're not sure
No.
which is in this case. So how could we distinguish? Is it belong to the abilities of the fungi or ability of the bacteria?
Yeah, that's a good.
AGF là gì chị?
So this is the streptomyces,
Yeah.
and it would appear that it is in this way.
Còn cái ít đó là của con streptom.
It would, if this is the streptomyces, it would appear that it cannot grow
Yeah.
towards the fungi. So probably the fungus is putting in the medium
Yeah.
antibacterial antibiotics actually.
Jan, do you have any questions?
No.